I would like to welcome you to a seminar in which you will learn about modeling, about what is Genome Enhancer and how you can use it to accelerate omics data, and how to build models to get most information from your data. During the development of the optogenerapi solution, geneXplain has been in charge of optimizing the optogenetic pathway to predict the behavior of the cells involved in the production of interferon beta and simulate different cascade scenarios within the engineered cells. Today, it's a pleasure for me to introduce Dr. Alexander Kerr, geneXplain Chief Scientific Officer. Hello, Alexander.
Hello, Beotza, and hello, all the participants. As Beotza has kindly introduced our company, so we are geneXplain company, which is a small company located in Germany. It's a small North German city called Wolfenbüttel, and you see some picture of it just on the wall behind me. So let's just start with our presentation of the project first of all, and our role in this project, and how geneXplain contributed with our software to analyze the data which was generated in this project. So basically, Optogenerapy is a very interesting and I would say high-tech project where special cells were engineered which are able to accept light. And when they accept a special type of light, then through this mechanism, certain genes can be switched on. So basically, certain constructs were introduced in these cells, which keep several genes, like genes encoding proteins, which are able to get the light. And then through the signal cascade, which is shown here in the picture, this information, this signal from the light is going down through the cell into the nucleus, where a promoter of a gene, which has to encode interferon beta, is expressed, and then this product is used to heal the basically to multiple sclerosis. So these kind of cells, then they put in a special device which could be switched on remotely. So you see, it's really high-tech stuff. But of course, at the first step, this whole construct was done in vitro, and our goal was to check what happens with those cells if we start, activating them with the light. Basically, after light activation, these cascades start to work. And of course, first of all, we have to see that the protein which we want to express is going to be expressed. But other stuff would happen because actually the cells are alive and they will receive the signal, and certain transcription factor, which is called IRF3, will be phosphorylated, and consequently all the genes which are able to receive this signal will be also expressed. And basically these cells may become unstable because of that stimulation. So our goal was to see what could happen with those cells. And for that, in our consortium, the partners made an experiment. They basically induced the cells with this NIR, so this light. And independently, as a control, there were cells, the same cells which were growing in the dark without the light. And then after 2.5 hours, the expression of the genes which were observed in the induced cells were compared with expression of the genes which were actually expressed in the cells which are growing on the dark. And then while doing this comparison, we hope to see what's happened with those cells and what kind of processes are actually activated in the cells. And to analyze this, we use our software called geneXplain platform. And this software is pretty big, so it has a lot of tools to analyze different types of omics data. Basically, this platform was developed to analyze many kinds of omics data. We were talking today about RNA-seq data, but actually we can analyze with this software proteomics data, epigenomics data like CG DNA methylation, ChIP-seq data like, for instance, ChIP-seq related to transcription factors or status of chromatin, microRNA data, and many other types of data. So, but today we will apply this tool for analysis of RNA-seq data, which was generated in this project. So the data first were loaded, and basically, if you click on this RNA-seq button in the screen, you get a list of possible pipelines which this tool can perform to analyze the data. First, RNA-seq data were loaded as a FASTQ files, so the raw format, and they had to be preprocessed. Basically, the FASTQ files were aligned to the genome, and the counts were done. So basically, we know after that which genes are upregulated or downregulated. And for that, the number of pipelines here to provided to calculate the up or down regulated genes. So what we did, and basically after that, after calculating the upregulating and downregulated genes, we want to know why they are actually upregulated or downregulated. So, and we know that expression of the genes is controlled by transcription factors. And the transcription factors are actually binding to promoters of the genes and switching them on or off. And the very important part of our platform is to possibility to analyze the promoters of the genes. So, for instance, in this case, we have a number of genes which are upregulated, so-called differentially expressed genes, and we have a number of genes which didn't change expression. So we call them control genes or no set. And then each line here is one promoter, and then our tool would start searching for transcription factor binding sites for all kinds of transcription factors. For instance, transcription factor NF-kappa B. And with these tools, we can see how many sites of NF-kappa B we can observe in promoters of upregulated genes, and we can count how many of those sites we can observe in control genes. And then this tool can compare the frequency of the sites in the promoters of upregulated genes and promoters of non-change genes. And then if you see that this yes to no ratio, basically we see that 4 times in this case, in this example case, the sites for NF-kappa B are more frequent than in the promoters of upregulated genes. We can also calculate p-value for that. So with this method, we can analyze now our gene list. How it is done? Basically, as you remember, we are dealing now with transcription factor, which is called IRF3. So this is a transcription factor which was introduced in our cells and has to be activated upon activation of the cell by the lights. So we know how these transcription factors are binding to DNA. So we collect and transfect various types of matrices, so-called positional weight matrices, which help us to find these binding sites in the new promoters, like it is shown in this picture. But actually, what is really important to understand that transcription factors like IRF1 or other types of factors, they are very rarely actually acting alone. In most of the cases, they acting in combinations with other factors. So for instance, NF-kappa B, which we mentioned already today, has to interact with IRF3 sometimes, or maybe in other cases, other factors have to interact with IRF3 in order to activate the genes. And these combinations of factors has to be found in the promoters of the upregulated genes. And we call them composite modules. And our platform has a special algorithm which is able to find these composite modules. It's applied certain machine learning techniques called genetic algorithms, and it searches for combinations of binding sites in a particular local area around the promoters. So I'm not going into much in details how it is really done, but at the end, the tool will return to you once you give a set of promoters, for instance, promoters of these upregulated genes. It will return to you a combination of transcription factors, like it is shown on the screen, like these 10 transcription factors tend to be found, the binding sites for these 10 factors tend to be found in the local area of the promoter, of all the promoters on the study. So it's pictures like that we'll see. So this, in this case, it's an example model which can be found. And then the next step in our analysis is called Upstream Analysis schema. So basically, those transcription factors which will be found to be responsible for upregulation of the genes, then will be subject to a further study. So basically, we will ask a question whether in the cells there are some master regulators in the signal transduction pathways which may simultaneously activate those transcription factors which we found. And this is done by a special algorithm which goes through the signal transduction network, which is taken from our database TransPath, and it searches short passes to this network from transcription factors, which are blue, here to the key regulators or master regulators, which are red here on the schema. So this master regulator is able to actually transfer the signals through the signal cascade and this signal is able to reach these transcription factors of ours in our study. So we basically applied this algorithm to the upregulated genes, to the data which we loaded in the system, and we detected not just one gene is upregulated, but many genes. Not so many, I would say, but around 300. And you see here a set of top 10 upregulated genes and top 10 downregulated genes on the schema, on the screen. And among them, you see very interesting proteins and genes which are upregulated. But of course, to understand what is going on, it's just to look at the list is not enough. So you have to really go further, first to analyze promoters of these genes. But before that, we even our platform has all kind of tools to analyze basically what these genes are doing. So it was interesting to see that many of those genes are actually express proteins which are targeting the Golgi apparatus. And they also important in translation. So it's very basic mechanism which are affected by this light in this case. So basically IRF3 is activated and then many genes are upregulated and you see their very basic mechanism are affected. So when we look at the signal transduction canonical pathways which are affected, we see that such pathways like pathways related to interferon gamma activation or IHR pathway, they're actually downregulated. Interesting. We also can see which types of markers of different diseases, human diseases, are also acting here. So after activating all those cells. But when we look at really inside, the top pathways which were in fact upregulated were pathways related to ubiquitination and special type of ubiquitination which is involved with such pathways which are called involve protein parking. And on this schema, you see results of gene set enrichment analysis where the upregulated genes are located in this scale at the left side of the diagram, and this red arrow show how many genes from this pathway. There are two, and then few of them next after that. So they are really located on this diagram at the left side. So it shows that these genes are really upregulated and they belong to this pathway. So parking pathways was important. And when we look at the promoters of those genes, we revealed this combination of transcription factors. And among them, of course, we see the IRF factors. So you see this IRF9 matrix, which is the positional weight matrices, which is recognized, showing the binding sites for IRF factors. And there are also other transcription factors which seem to be interacting with these factors in the promoters of our genes. And you see those like AP4 and P53, by the way, and ETS factors. And they put together, made such a combination, which is then discriminate promoters of upregulated genes from the promoters of non-changed genes. And then the next step was to actually to look the upstream from these transcription factors. And to our surprise, we see the only small network, which is really, in this case, was picked up by the system with a keynote, which was a heat shock protein. And this heat shock protein was actually downregulated. And through this downregulation, it transmits the signal to those transcription factors, which we reveal in our analysis. So why this heat shock is so important? If we look at the heat shock protein of this type of heat shock protein, simply in Wikipedia, we see that it's really a very important protein. But what is interesting for us, it's in fact is involved in inhibiting apoptosis, and that's actually very important because our main worry was that this construct which we created may be sensitive, and those cells, after induction by the light, they will start dying, for instance, which we don't observe. But we also do observe that certain pathways are activated, which in fact inhibit apoptosis. So those cells are going to live longer, which is a good news. Actually, in general, there are also reports showing that this ubiquitation pathway and parking pathways are already observed in the cells which are where stink trafficking is actually activated, and that's exactly what we are doing in those cells. So basically, we see now details of the mechanisms which are going on in the cells, and further on to understand what will happen with the cells after in many hours, and actually hopefully in days and maybe months. And to be able to predict what will happen with the cells, we then will start actually modeling using our pathways which we detected. But now I want to introduce to you actually a new tool which we come up recently in our company, which is called Genome Enhancer. And Genome Enhancer is realizing those steps which I just showed to you, but it's doing it completely automatic. So, actually the idea of this tool is like an automatic car. So basically, of course, you can drive your car with all kind of tools, like we have geneXplain platform if you want to drive it to a particular direction. So you really have to have your care. But if you want to really to go further with an automatic procedure, which we often want to do, so for that purpose, we created a pipeline where you simply put your data, you click one button, and then you get the result about the pathways which we detected, about the master regulators which we predicted in the cells. And we even gone further. if we analyze disease data, we want to actually come up with a disease mechanism and essentially, eventually also with potential drug therapies against those diseases. So I want to demonstrate how does it work. So it's a web tool. Once you get the account in this tool, you open it and you get such a drop box actually on the screen where you can put all your kind of data. And here you can put really all type of omics data. You can put RNA-seq data, you can put proteomics data, you can put even metabolomics data, and the system will automatically understand what to do with this data. So you simply drop your files in the screen. You can also load them from remotely from some FTP site, and then you get such colorful boxes. We like the colorful boxes, and you would put your data in different categories. In this case, I want to show you an example of actually data coming from a disease which is related with inflammation. So actually, IRF3 transcription factor, which we are studying here in this project, is always related to different types of inflammatory processes. And here is an example. I want to show you analysis results of a disease called IBD, inflammatory bowel disease, where actually in our gut of some patients, inflammation for some reason is going on. And we want to understand the mechanism of this inflammation and actually to find potential drugs. And so these data were generated not with RNA-seq, but with microarrays. But our tool is able to deal with both with RNA-seq and all kind of microarrays. So basically, we get here inflamed and non-inflamed tissue, and the data were taken from inflamed and compared with non-inflamed. Then the next step in the tool is to specify your disease. So there's a big list of different diseases in our tool, and you select the disease you're dealing with, inflammatory bowel disease, and you specify which conditions you want to compare, like inflamed versus non-inflamed. And then you start the analysis. You click the big button in the center of the screen. Then it starts running. It takes several hours to finish, and at the end, it generates a complete report. And this report is looks like a kind of example of a scientific paper. Basically, first it takes this data, it discovers differentially expressed genes, and then it starts actually analyzing it. It basically then plot it in the form of heat maps. It then start to see which kind of geoterms are activated, overrepresented among those up or down regulated genes. Like in this case, you see, for instance, that of course a lot of immune disease genes are activated here. It also look at the pathways which are activated, the canonical pathways, and you see, for instance, in the case of IBD, interleukin 8 pathways is activated, and other important pathways. It also look at the known biomarkers of different diseases, and you see here, for instance, the top heap here is immune system diseases. So obviously, a lot of genes which were activated in this experiment, or in this, actually not experiment, but in this tissues, inflamed tissues, belong to immune system diseases. But then we were also able to, in this case, to add proteomics data. So, and then system knows, okay, if we have transcriptomics and proteomics data together, we can compare it. So what it did, it also compared upregulated genes in transcriptomics with upregulated in proteomics. And then it did similar analysis which I presented in our case. So we search for in promoters of those genes, revealed composite modules which are enriched in promoters of upregulated genes in inflammation. Then they start building upstream and searching for master regulators. And you see this network which was built by the system automatically, and it shows which master regulators, which are shown here in red, can activate the transcription factors which are shown here in the blue in our system. And basically with highlighting, we also see which of these proteins are upregulated in proteomics data or in transcriptomics data or in both. So it, for instance, STAT3 is activated in observed, upregulated in transcriptomics, and also observed upregulated in proteomics data, although other proteins may be only proteomics or transcriptomics are observed. So both types of data are taken together in this analysis. And then next step, which this system is doing automatically, is actually looking in our database, which is called human PSD, and it checks which drugs, known drugs on the market, or those drugs which are going on through the different types of clinical phase. They are actually known to be inhibiting those targets which are found among our upregulated genes and which are found among our master regulators. So, and then it prioritizing those drugs and give this list of drugs as a potential drugs which can be used for the therapy. This can be done, of course, for the big set of patients, but also individually for each individual patient. This system will then prioritize the drugs accordingly and suggest it for this particular patient. It also, for the drug development studies, it also proposes certain active chemical compounds which potentially may also downregulate or inhibit our master regulators. So this is done based on chemoinformatics tools. And one of that, which is really very interesting, is called PAS. And it's based on the chemoinformatics principles. So at the end, this tool generates automatically such a schema, where it shows which master regulators were found in our data, which transcription factors are actually controlled by those master regulators, and which drugs may be used here to control master regulators. And it gives also all kind of references and methods section, which describes what types of methods were used. So basically, with this, I want to finish and give you possibility also to ask some questions. But here I demonstrated how our tools helps to understand molecular mechanism of complex processes which can happen in the cell. They can also help to understand the molecular mechanism of diseases and even propose different drugs against those molecular mechanisms revealed in the diseases. So with that, I want to thank you, and I open for the questions. Okay, so until you are ready with your questions, I can show a bit of the tool online. So let me again share my screen. To be able to analyze your data, you go just simply to geneXplain.com, and here there is a description of our products, and you can read about our main products like TransFac and TransPass. These are important databases which I mentioned today, geneXplain platform or Genome Enhancer. So if you want to log in, if you have already your login account, you can directly log in, or you can get an account. So for instance, if you want to have an account for Genome Enhancer, you click here, and here the register button. So with the register button, you'll get at this page where you can register. There are several actually possibilities to register. You can get accounts for 1 week or for 1 year, or you can get actually a free account where you can, for 1 day. Well, 1 day you can trial the whole system. Of course, the account will stay for the whole time, but just to try the system actively for free, you can do it for 1 day, or you contact us to get maybe extended trials, or if you need to have more ideas how to do that. So once you have your account, then you go to the system directly, like I did. So basically, let me show you. So I have my account, I sign in. And now here in the account, there's several possibilities. First is you can start a new project by clicking on Start New Project button. We'll do it maybe in a moment, but first you can look at the existing projects. So there are some demo projects which you can actually see what is about, and you can also look at the results of those projects. But first I want to maybe demonstrate how this project works. So this Start New Project, it will open for you basically such a drop file. And here I want to show you example of one project where I analyzed actually multiple sclerosis data. And I took it from very recent publication, which you see on your screen, where actually 106 samples from brain were analyzed, some of them taken from post-mortem, of course, multiple sclerosis patients, and some of them from control. So I took this data, it's actually, you can load them through the FTP, and drop them here. So drag and drop this text file here. After that, you simply press next, and you'll get such a gray box where you have to put your data by drag and drop it here. So it will add this to this gray box. Then you play next. So actually you want to work with this data. Now you need to specify which of the samples belong to category multiple sclerosis, and which of the samples belong to the control. So you, again, by drag and drop, you specify which samples belong to what. And then you press next. And here is this, the only interface you have to work with. Basically, you have to select the disease. So you type for instance, multiple sclerosis, and you see how many other diseases are present here. And then you select conditions you want to work with. So you select multiple sclerosis gray matter samples versus control. You can specify the tissue where actually the expression was measured. In this case, it was brain. And then you simply start analysis. And you see it is now started. And in fact, I did it already. So it took about 4 hours. And then it generated these two stories. We call it brief tale and full story. Let's click on full story. It's now opening it. So it created such a report where this title of the report is automatically generated, which is saying PLK1 and FAS are promising druggable targets for treating multiple sclerosis that control activity of these transcription factors in promoters of differentially expressed genes in brain tissue. So then it create an abstract, an introduction, and it shows which kind of data were used. So you see this gray matter versus control. Then it identifies differentially expressed genes. You see upregulated genes, downregulated genes. And so there is a question, but let me just finish. You see there is basically heat maps and geoterms, pathways, which were detected there. And then it goes for downregulated genes. Then we see which transcription factors are upregulated, which are found, in the promoters of upregulated genes. And you see, for instance, IRF1 here again, and STAT. These are the transcription factors which were found, again, for the downregulated genes. Now, it goes into the section of master regulator search. So it identifies those master regulators for upregulated genes and for downregulated genes. And it shows these networks. For the upregulated, you see these ones which are related to actually interleukin-1 pathway, and the downregulated also here. Then it searches for the potential drugs which can be used here against the upregulated master regulators. And they are sorted according to the several criteria. For instance, those drugs which can be targeting our found targets and which are already in different clinical phases against multiple sclerosis. And you see some of the drugs which you may know if you are working in that field. So you see the priority of those drugs is according to, as I said, multiple criteria, how well it is tested, and also how many of the targets from my list are potentially inhibited by this drug. Okay, so I think I have a question, so I have to look at another screen. So I want to write the question here. It is possible to also to integrate non-omics data into the analysis. For instance, survival data or no, not yet, not really in the full extent. Basically, these clinical data are modeled by those categories which are presented here. So actually, for instance, in this case, so there are patients. If you have different types of patients, for instance, if you have clinical data about short survival, long survival after certain treatment, or different types of types of patients which is important for your diseases. So what you will do, you will create new categories. So like I click here, so you create a new category, for instance, a short survival. And then you put all your data in this category, and then you will use this category in your comparison. So it's actually a little bit of manual work, and but that's this way you can involve basically this clinical data. The survival data, so we are now planning to use it automatically. It's important for instance, in case of cancer, which is very often used. Our system is very often used to analyze cancer data, and for the cancer type of data, we will also use the survival information. So the question, is it possible to handle more than two conditions for a disease? For a data set, right. So as I mentioned here, so you create two conditions, so you create more conditions. For instance, as you can see in this case, I created a third condition, which was a certain cluster of patients. So I did a clusterization of patients, and this is a cluster of a very special cluster of patients which behave a little bit different from others. So now I have actually 1, 2, 3 conditions, and then I can either compare two conditions to each other. So I would say I want to compare this cluster to the control, Or I could analyze several conditions together. For that, I create more conditions here. And I will then create, actually, say, to take all the conditions, like MS cluster 1, the total MS, and then I will select one of them as a baseline, which means all of these conditions will be always compared to the baseline. If I'm not selecting the baseline, then actually what will happen, the system will take those conditions together and first will cluster them and find basically the baseline by itself. So we try to put a lot of artificial intelligence in the system so that it makes decisions by itself if you don't want to do it by yourself. If you can do such decisions yourself, then it obeys your decision. Is there a minimum amount of information, informatics resources that are allocated for each query, or did it took 4 hours just because you are the boss? No, it's actually all related to the hardware which is running behind the scene. So our tool is installed on a server here, not far from here, about several hundred kilometers, in a data center. And we have quite a powerful server with many cores. And then depending on how many active users are currently running on the system, it will share the resources accordingly. It's never giving all resources to one user. It's actually always keeps some users, some resources free for users to come. So, and 4 hours, it's average time. So of course, if the server is too busy, maybe it will take longer. And also depending on type of data you have. For instance, if you have raw data like FASTQ files, then the system will take additional time to do the alignments, to all these kind of steps, which takes time. If you have already differentially expressed genes done, so what you do is just load your list of genes, and then for the system it will be easier, and it will take a short time. Another question. In some composite modules cases, false positives and false negative values are more than 40%. I would like to know, is there any false positive and negative cutoff for good composite modules? A very good question, and very good observation, actually. Very often these distributions of values, like, let me just show here on the screen, but in this case, are quite overlapping. So what we see here is this score, which is basically frequency or power or efficiency of that composite module in a particular promoter. And the higher the score, the better is the composite module in the promoter. And you see the promoters of upregulated genes are red and the non-changed are blue. Although they are shifted, and they are quite significantly shifted, so you see the Wilcoxon p-value is pretty significant, still there is an overlap, and this overlap can be quite big. And which gives actually important message. The data are really noisy, and the noise comes from many different, many different sites. For instance, we actually don't know which part of genome is active. So we propose that these are promoters which are actually regulating the genes. But in many cases, we are wrong. In fact, if we would have data about open chromatin, and sometimes you have it, these distributions will look much better because we will know exactly which region of genome is open and involved in regulating the genes. Here, we just take the promoters of the genes, and maybe in 40% of the cases, or maybe more, we simply are wrong because we don't know which region of genome is actually regulating my genes in this particular case. But still, although it's noisy, we find a significant shift. So it gives us good evidence about this composition of transcription factors which are involved in regulation of our genes. And which cutoffs to use? Actually, behind this scene in this tool, we check if the this AUC value is usually you know this is a value which demonstrates how good is your recognition function. If this AUC value is high enough, and if it is significant. If we found that it's not significant, then the tool will report that sorry, we were not able to find any cluster which would discriminate upregulated genes from downregulated genes, and it will not report results. So we have an internal threshold which is based on statistical significance. Okay. What is the minimum number of replicates required for RNA-seq analysis, and is data from the mouse model can be also analyzed? A very good question. The more the better. So the more number of replicates, the better. But we have very good results even if you work with few replicates. Actually, the minimum, technical minimum is 2. If you have 1, then the tool will also work, but it will not calculate significance. It will simply rank the data according to the values and take the top 300 genes from this list based simply on the value you are dealing with. For example, intensity of a signal or fold change, which is computed by just intensity from one sample. But if there are at least 2 samples, it will try to estimate the statistical significance. With 3 is a kind of okay. And of course, if you have more, 5, 10, 20, then it's really good. But of course, it all depends on how noisy is your data. For instance, in very often diseases, definition of a disease is so vague that you get samples which are really heterogeneous. And then even if you have 100 samples, the noise is so high, so it will not find a statistically significant differentially expressed genes. And in such cases, you better first cluster your genes, your sets in various types of clustering algorithms and define subtypes inside your molecular subtypes inside your definition of a disease. And whether we can analyze mouse data, yes and no. Yes, of course we can, but in the platform tool. So the GeneXplain platform tool, which I showed just in the beginning of my talk, is able to analyze human, mouse, and rat data. The Genome Enhancer, this automatic pipeline we just showed, dealing so far only with human data. So we are working on mouse data as well, but it's still in progress. Okay. Okay, there's no more questions so far. I guess we will finish with that. So we are already almost 2 hours, 1 hour. Yeah, 1 hour is fine. Yeah, okay. So, but if you have any more questions, you have our emails, right? So you can send the questions to us concerning everything what you heard today about all other tools and products we have in our pipeline and our portfolio. Also questions about prices or regulations of how you can get trial to analyze your data first and to check what you can do. So please, we are open to any questions and we'll answer promptly. Okay, so with that, I would like to thank you that you participated, and Bjornsa, do you have anything to add?
Yeah, I wanted to say thank you very much for GeneXplain for this complete presentation, and also for showing off some of the results of the project, and I guess particularly for showing how important is this modeling when you are talking about cell-based systems. I hope you have enjoyed this webinar, and also if you want to know a little bit more about our project, you can check our website at use.optogenerapy.eu. And thank you very much all for your attention.
Thank you very much for your attention, and goodbye.